Dr. Inoue is an evolutionary and conservation biologist with Shedd Aquarium's Daniel P. Harther Center for Conservation and Research. His study subjects, freshwater mussels, are among the most imperiled group of organisms in the world. Dr. Inoue leads freshwater mussel research in the Great Lakes region to understand how human activities and environmental change affect current mussel diversity. Before coming to Shedd in 2019, Dr. Inoue was a research assistant at Texas A&M, where he used genetic analysis to assess conservation status of freshwater mussels in Texas. Prior to that, he was a postdoctoral researcher at the Technical University of Munich, Germany, where he worked on European freshwater mussels and fishes. And with that, I would like to turn things over to Dr. Inoue.
Thank you. Thank you for a warm introduction, and I'm glad to be here. Today, I want to talk to you about current ongoing research at Shedd Aquarium and what we are doing, and then how we can use our research to conserve this most imperiled species in the world. So I want to start with this. Most of you might not know that Shedd Aquarium has actual active research program. So we actually have 10 PhD researchers dedicated to work on specific organisms and then try to conserve species. So it's from marine system, from marine iguana to corals, sharks, to freshwater research, such as amphibians, migratory fish, and freshwater mussels. And then, so today I wanna talk to you about the freshwater mussels and then my research program at Shedd Aquarium. So I started a Shedd Aquarium job as research biologist in 2019. So this is my third year. And then since then I started field research and lab research. So my talk today is just portion of field research and genetic research that I'm currently working on, and some of the results. So most of you might not know what is freshwater mussels. So I want to start with kind of small introduction to freshwater mussels. So these are freshwater mussels. I took pictures over my research career. So we have about 300 species in North America and the United States. I want to talk to you about a little bit about what they do in the ecosystem, and they provide essential role in the ecosystem. And then freshwater mussels are one of the most endangered group of animals in the world. And so next few slides, I want to talk about what they do and why they are so endangered. So they provide essential roles in the ecosystem, and then they are basically filter feeders. They filter water constantly, basically taking nutrients. So they eat bacteria, algae, and even viruses and consume. So basically they are removing any material floating in the river. So they might be removing the bacteria, harmful bacteria such as E. coli. So they are cleaning water for us. And their body is good food resources for fish, bird, and mammals. A lot of mammals actually eat freshwater mussels such as raccoons and muskrat. Some fish can eat those mussels too. And while taking nutrients from water, they release excess nutrient through basically pee and poop. Those nutrients are very variable for microbes such as algae and other bacteria. And then their physical appearance in the river system is actually provide habitat for other animals. So basically they are freshwater mussels are free living bivalves, so not similar to zebra mussels. So they live in the river bottom. So they actually create a physical structure in the river bottom. They are stabilizing river bottom. But also once they die, they leave their shells in the system, in the ecosystem, those are also good shelters for other organisms. So often you can find the caddisfly nested on a mussel shell. And because they are water filter feeders and they live in a river bottom, they are very sensitive to environmental change, such as water pollution or habitat destruction and degradation. So they are good bioindicators. So if you see mussels in a river, that river may be good condition compared to no mussels river. And one of the very interesting thing about freshwater mussels is they have very complex life cycle. So like I said, most of the mussels are free living organisms in the river. So they burrow into the river bottom, such as this picture. And then they usually live close to each other. So they create kind of a dense aggregate, often called mussel beds. And then most of species can live more than 10 years. So they have very long lifespan. So that some species can live 10 to 100 years. And they have very unique life history. So this is a kind of simplified version of a diagram of mussel life history. Basically, I started with number one. So this is a male and this is a female. Male mussel release sperm into the water column, and female mussel intake those sperm through filtering water, and they fertilize eggs. Once egg is matured, the larvae, or we call glaucidia, they has to attach on fish host to metamorphose to juveniles. So the freshwater mussel larvae is actually the obligate parasites of fish. They usually attach on fish gills, but some species attach on fish fins or the scales. And they usually stay on a host about a week to a couple months, depending on species. And once the larvae grow to the juvenile, they metamorphosize, and they are dropped off from the host, and they are going to be free living mussels. So freshwater mussel require fish to complete life cycle. And then usually the one female produce 20,000 larvae to 5 million larvae, but only small fraction of them will survive to the adult. And because they are obligate parasites and then they have to attach on, the larvae has to attach on fish. So a lot of species, mussel species, use many different strategies to attract fish to make sure those larvae attach on fish. So this is one of kind of most popular, famous mussel display. So this was taken in the DePage River. But so this is a female, and a female mantle tissue is mimicking like a small fish, so that fish try to eat that small the fish, and then this female release glaucidia on a fish to make sure those glaucidia attach on a fish. And then this particular mussel species called plain pocketbook use a predator fish like bass and sunfish. So they evolved to shape like small, small prey item, so that those predator will try to attack that small fish and then make sure the female release the glaucidia and then touch on the fish. And then there are a lot of strategy to attract host fish. And then many mussel species are host specialists. This means that some species can only use one or few fish species to complete life cycle. So, so I put 4 different strategies here. Like some species actually catch fish and then make sure the glaucidia is released on the fish. Some species make like crayfish-like display. This species is very interesting. They release like small packet of Glaucidia, but it looks like little worm. So that small fish like darters try to eat that prey item, and then the Glaucidia is going to attach on darter's face and then gills. And then this is, last one is a kind of very special kind of strategy. They actually fish, using fly fishing kind of method. So this is kind of modified tube formed by the mantle, but tip of mantle we have a packet of the Glaucidia, so that fish try to eat them and then we break down. So if you are interested in those things, I highly recommend to go to Unio Gallery. Dr. Chris Barnhart at Missouri State University, he put a lot of pictures and videos, and it's very interesting. But one of this life history, like this attraction of fish, that makes those freshwater mussels more vulnerable to the environmental change, because they need to have fish, and if environment change and if fish is gone, they cannot really reproduce. So where we can find freshwater mussels? So we basically can find freshwater mussels pretty much everywhere, all continents except Antarctica. And then they live in a big stream to small creeks. So this is a kind of extreme place I've been. So like Rio Grande, the border of Mexico and US. We went to Newfoundland, Canada, and then we found some mussels. I'm from Japan, so we also have mussels in Japan, and then Mexico, Germany, and then this is upstream of Mississippi River. So basically, we can find freshwater mussels pretty much everywhere in the streams. But like I said, freshwater mussels are most endangered group of animals. And then they are currently experiencing severe declines in population size, and then a lot of species are going to be extinct because they are very sensitive to environmental change. So the water pollution, habitat degradation, flow modification, invasive species, more recently, disease caused by virus, all makes those species and populations to decline. So we have about 300 species in North America, but about 10% of those species already extinct. And a few weeks ago, Fish and Water Service just announced that 8 species are declared to be extinct. And the remaining 65% of species are either threatened or endangered, listed by state or federal level in the United States. And especially in the Chicago and Midwest region, these three are one of those are big component for those species to decline because we have higher human population density and urbanization and industrialization, but also dominant landscape around here. Land use right here is agriculture, and then there are a lot of out at the runoff from agricultural field. So in Shedd Aquarium, I'm working on freshwater mussel research and conservation. And then I'm basically doing one part feed research. So try to document local species richness and distribution around Chicago and northern Illinois, and monitoring the health of mussel communities over time. And I'm going to present this in the next few slides. Another research I'm working on is genetic research. I'm trying to understand species diversity and taxonomy and evolution using genetic tools, but also I'm trying to understand more species level genetic diversities, and then how spatially those genetic diversities are located. So try to understand genetic diversity amongst populations and how those populations are connected by gene flow. And we try to use those kind of research findings to basically make better management and conservation for freshwater mussels and their habitat. So next few slides, I'm going to share my ongoing research on mussel community monitoring. And the goal is try to understand health and stability of mussel communities in the Chicago region over time. So more specifically, we are looking at to monitor changes in community composition and population dynamics over time, and try to determine the effect of environmental factors such as climate, like water temperature or air temperature, precipitation, and flood and drought on mussel survival, and how those environmental factors change over time. So for those studies, we have 3 monitoring sites in the northeastern Illinois. So one site is North Branch Nippersink in McHenry County. The other side is in the South Branch Kishwaukee River in DeKalb County. And finally, we have Poplar Creek in Cook County. So, and we are collaborating with Open Lands in Nippersink Creek, DeKalb County Forest Preserve in the Kishwaukee River, and the Cook County Forest Preserve in Poplar Creek. And we are using, it's called mark recapture monitoring. So basically going to the site and capture all animals in that site, and then mark them with tags. So in our case, we put some unique number vinyl tag on the mussel shell, but also we put the pit tags, which is kind of similar to microchip in your pets, like cat and dogs, to identify animals. So we put those tags on mussels and then release them back to the same habitat. And then we repeat that process over time. So we basically track each individuals over time. So we know which individuals survive 1 year to next year. But at the same time, if animals found in a site is not tagged, that individual may be migrated from outside of the site, or they born in a site. So we can understand survivorship of the mussels, but also recruitment and migration of the mussels in the site. So try to understand demography and population dynamics. And at the site, we are also collecting some environmental variables, such as river discharge and water temperature. So at each site, we have designated plot. It's 150-meter square plot in each site. And basically find, like I say, we find all mussels in the site and then tag with vinyl tag, like each tag has a unique number on it. And then release them to the same site, and then recapture next year, and then to find out what we have in the next year. So we established the mark recapture monitoring sites in 2020. And in 2020, we tagged total 644 mussels over 16 species. And this is the number we have in each site. So Nippersink Creek has less individuals in there. So we have 96 individuals in that creek. But South Branch Kishwaukee River, we have 413 individuals tagged in 2020. And we did same monitoring this year in 2021. We recaptured total 493 tagged mussels over 3 sites, which makes a recapture rate of 76.6%. And we also tagged 268 new mussels. So total, we've tagged 912 mussels over 2 years. And it's very interesting that most of sites, all sites, population sites actually increase. But the Nipperson Creek has low recapture rate. Only 36.5% of animals we found this year, 2021, are tagged. And rest of them are actually new mussels, maybe came from outside of a plot, or they are reproduced in a plot. But Poplar Creek and in the South Branch Kishwaukee River, we have high recapture rate over 80%. So most of the mussel we found this year was actually tagged last year. So over the next few years, I want to know this trend stay as same or maybe changing over time so that we can understand population dynamics of that mussel community over time. So this is just a diagram of what species we found and how many we found. So we found about, so we found 10 species in North Branch, Kishwaukee, and Nipperson Creek, 5 species in Poplar Creek, and 12 species in Kishwaukee River. And then so this is basically the proportion or absolute abundance of the mussel species. And like you see, 2 sites have the higher abundance of muckets. So Nipperson Creek and then Kishwaukee River has mucket dominated, but Poplar Creek has ellipse mussels. And if you look at the proportional differences, in between years, we kind of see similar patterns of the proportional change. So the mucket is always abundant in a Nipperson Creek in both 2020 and 2021. And then in the Kishwaukee River, most of the muckets are found 2021 is tagged. So it's not really changing much. And then in a Poplar Creek, it's a different species. Ellipse is most dominant species. It's about 95% of all found ellipse species. The species is ellipse. But it's interesting that the Nipperson Creek has more wide variety of species, has a higher proportion of species abundance, but the rest of sites has one very dominant species, and the rest of them are kind of small, more rare species. So based on 2 years of monitoring, I can say some the significant species. So like I said, the mucket is most abundant in the Nipperson Creek and Kishwaukee River. This species is very common around Midwest rivers and streams. The next significant species is ellipse. This species is a species of special concern. So it's not listed under federal protection or state protection, but we are considering this species is next to the listing. This species is widely distributed across Midwest states, but it's very sporadic and in very limited locations. They can find in limited locations, but it's very abundant in the Poplar Creek. So 91.4% of individuals we found are ellipse. And black sunshell is also kind of significant species. It's formerly state threatened species in Illinois, currently species of special concern. It's relatively rare and usually found in the mid to large rivers. We found one individual in the North Branch Nipperson Creek. It's a small creek, so it's kind of surprising. We tagged one and then we are monitoring this. Fluted shell is also a species of special concern, widely distributed, but relatively rare in the northern states. But this species was fairly abundant in Kishwaukee River and the Nipperson Creek. And lastly, it's called elktail, which is also a species of special concern, widely distributed across United States, but usually we can find this species in a small number of individuals, never be abundant. but we tagged 12 individuals across sites. So we keep eye on those species, and then we will see how those community composition change over time. So basically, this monitoring work is gathering baseline information so that we try to understand the current condition of freshwater mussel community in those sites, and understand, try to create kind of conservation strategies for those sites. So what we found is we found high recapture rates, but not one site. So Nipperson Creek has a low recapture rate. I want to know this is constant over time. The population size was actually constant or even increasing in all sites. And we also found a high variability among sites. So species composition and abundance are pretty different across sites. So this is based on the 2 years monitoring data. So we obviously need to have more long-term monitoring data to examine what the temporal trends in those sites. So in summer 2022, we will return to the site for the 3rd year monitoring. And at that point, at least we have 3 years monitoring data. So we will analyze some changes in demography and in species composition at the site over 3 years, and examine association between demographic parameters, survivals and recruitment and so on, and environment. So water temperature and hydrology. And ultimately, we will evaluate potential conservation actions at each site based on our monitoring data. So this is ongoing, and then this is part of field work. And the next few slides, I want to share about some of the genetic research we are working on. So conservation research, conservation genetic research project. And in this kind of project, we try to understand how genetic diversity, so I try to understand genetic diversity of natural populations and how those genetic diversities persist long term. So specifically, we have a couple of questions for this project. So one is, how is genetic variation distributed within species? And then what happens to those genetic variations as population size decline or populations isolate from each other? So there is no gene flow, or populations are declining, so genetic diversity may be reduced. And then particularly for the next few slides, I'm going to talk about a project that I worked on dealing with a captive propagation project. And then we try to understand how does population captive propagation affect genetic makeup of juveniles and wild populations. So as mussel populations are declining, propagation, augmentation, and reintroductions have become major action, conservation action for species recovery. This is because during the life history of freshwater mussels, the transition from Glaucidia to juveniles is always hindering, because they require fish to metamorphose to juveniles, but at the same time, very small proportion of Glaucidia becomes to juveniles, and then again, those small proportion of juveniles will survive to adult. So those in those steps, we can actually help to increase the number of juveniles by captively propagate, and then subsequently release those propagated juveniles to the natural habitat by augmenting the existing populations or reintroducing to the site that mussels used to be there, but then currently gone. So the propagation, augmentation, and reintroduction is very popular conservation actions currently doing for the species recovery. But there may be some negative genetic consequences if we are not carefully working on propagation. Because if you're propagating from single female or very few number of female, those offspring may be all related. So maybe we are reducing the genetic diversity, maybe increasing the inbreeding by propagating from a few number of females and releasing those large number of juveniles in the natural habitat. The other consequences maybe is those artificial condition control environment may create kind of domestication of freshwater mussels. So maybe those juveniles, or luckily in juveniles, maybe adapt to the artificial environment so that they survive in the artificial environment, but they might not be able to survive in the natural condition. And then if we release a large number of individuals in one place, it may be misrepresenting or maybe even modifying local gene pool because we are taking from a few females, and then those offspring may have very similar genetic makeups. And then I want to bring this diagram back because in life history, the male release sperm in the water column, and a female capture those sperm. So female may be capturing more than one sperm, so sperm from more than one males. So there may be multiple paternity within the female and then within the offspring. So multiple paternity is common in the freshwater mussels. And then this potentially retain genetic diversity because even one single female, those offspring might have multiple male contributions. So they might be retaining genetic diversity and maybe reducing the inbreeding. But this whole life cycle makes difficult to predict. So in this study, we team up with DuPage County Forest Preserve District and I work on two common species, common mussel species. One is called plain pocketbook and the other is called fat mucket. And then we did kind of experiment followed by genetic analysis. So first we propagated juveniles from single female for each species. And then we also collected wild adults from the same place where the female was collected. And then we try to understand how much genetic variation is retained within the offspring from a single female, and then comparing two species. So we estimated number of fathers within offspring, and we compared genetic variation between those propagated juveniles and then wild populations so that we understand how much genetic diversities are retaining within the offspring from single female compared to wild population that female was collected from. So jump into the result. This was kind of surprising result to me because so we have kind of small sample size. We have 18 juveniles for plain pocketbook and 37 juveniles from fat mucket. But based on the genetic analysis, we actually found 13 different fathers within the offspring of plain pocketbook and 25 potential fathers in the fat mucket. So this percentage is basically showing 72% of 7 offsprings have different fathers. So this is actually surprising because it's very high levels of multiple paternity within a single brood. So the offspring is from that single female. Like I said, caveat is the small sample size. So actual number of fathers may be much higher. So we are kind of following up this research, adding more samples. And then next we try to compared genetic variation between propagated juveniles and the wild populations. And then we use two different genetic diversity indices. One is allelic richness. It's basically that how many genetic variants or genetic variation are in each group. And heterozygosity is another metric we use. It's basically the genetic diversity within individuals. So heterozygosity means you have a different copy of genes within single individuals. And then we also estimated inbreeding coefficient, which is the value of how much inbreeding is occurring in that group or how much outbreeding is occurring in the group. The positive number is showing the inbreeding and the negative number is showing outbreeding. And what we found is those red lines showing the significant result between propagated animals and then wild animals. So in both species, allelic richness is significantly different between propagated juveniles and wild individuals. And then both species showed propagated juveniles has less allelic richness compared to wild populations. But we didn't find any differences in heterozygosity between propagated and the wild. And then we found one significant result in a plain pocketbook in an inbreeding coefficient, but this is actually showing that propagated animals has less inbred than wild populations. So virtually this is showing that propagated juveniles doesn't cause the inbreeding. So comparing between propagated juveniles and wild populations, we found that the allelic richness is reduced in propagated animals, but we didn't see any difference in heterozygosity levels, and we didn't find increase in inbreeding. The offspring from single female likely retain high levels of heterozygosity and maybe prevent inbreeding, but reducing the total genetic variation. And then we look at more closely how those genetic makeups are changing between wild individual and juveniles. So this is looking at a different part of genome, and then the color represent genetic variants. So let's say this is green variant and this is orange variant in wild population, and then this is propagated population. And then we are looking at the proportion of those variants within those groups. So let's say this green variance in wild population is about like 80% 80% in the population. So if you compare between a wild population and propagated individuals, we actually see some reduction of proportion reduction in one variant, but maybe some variants is increased. But if you look at the more the variance rich region of a gene, we also saw some missing variants in the juveniles. And then we also found in a fat market. So some of them change in proportion, but some variants are actually gone. So this may be showing that the slight reduction of allelic richness between propagated animals and wild animals. The propagated animals or propagation from single female may be changing allele frequencies. So this is a variant frequencies. And maybe losing some rare alleles. And because this is from single mom, even though they have multiple fathers, they are kind of homogenizing genetic variation toward like maternal variation. And it and then those whole result is showing that they may be reducing adaptive potential because we are losing some variants that might be important to adapt when new environmental factors came in or something like that. So what we found through this study is we found that the offspring from single female has higher genetic diversity than expected. And this is due to many fathers. The offspring has many fathers, but lose some rare genetic variants. This may be because it's from single mom, and also maybe because we have small sample size. But the fact is we found some rare genetic variants are kind of lost. So the conclusion is we should use multiple females, and we need to be a little caution about propagating animals, and because we may be changing a genetic makeup of natural populations, or maybe we are misrepresenting genetic variation. So propagation should be like action of last resort. So if we need to, so we can use propagation to save from same species from the extinction, something like that. So for future studies, kind of using similar systems, I want to examine with larger sample size, both juveniles and wild. I want to see that result is actually sustaining over if you use a large sample size. We also want to know this what the variability among females. So if you collect multiple females from the same site, are they having same paternal contributions, or maybe one female has 100 of paternal contribution, but maybe one female is not. So we want to know what the variability among females. And then also we want to know variability among species. So some species has different life history traits. And then also we want to know like comparing rare species versus common species. And if rare species, it's just rare, so it may be less number of paternal contribution compared to common species. So these are currently ongoing projects. And then we also have multiple research projects currently going on. But I just want to show our conservation collaborators and partners, because conservation and even research, we cannot do research without collaborators and partners. So we are so glad working with those collaborators, and then we try to understand those freshwater mussel species and try to use that findings to save those animals in a habitat. So this is kind of take-home message that we are doing research with a holistic conservation approach, and then try to adapt those findings to more the effective conservation management. So we try to advance our understanding of freshwater mussels and apply best available information for conservation. and species recovery. And then, yeah, so thriving mussel populations equal healthy rivers for everyone. So we want to have healthy mussel population, and then we want to have healthy rivers for everyone. So with that, I want to thank you all for listening this talk. And if you have any questions, shoot me email. And if I have time, I will take any questions.
Thank you, Dr. Inoue, for this great presentation. Again, if anybody has any questions, you can feel free to unmute yourself and ask them now, or if you'd rather put it in the chat and I can read them out.
Hi, Dr. Inoue. This is Jessica Collier. I have a question for you. First of all, thank you for your presentation. It was really wonderful and I enjoyed listening to you. I found your suggestion about the genetics of propagated or reared mussels really interesting. And I'm glad that you're putting work into this, especially about the genetic diversity, the loss of genetic diversity in propagated mussels, because there are so many potential fathers that are contributing to one single female when it comes to the reproduction. We found something similar with lake sturgeon. And so when we do captive propagation of lake sturgeon, we try to aim to have a minimum of 5 male sperm donors per female egg donor to increase genetic diversity. And your recommendation was that captive propagation for mussel restoration should be kind of a last resort. Do you think that there could be some kind of guidance in the future that, you know, I'm hearing more conversations that lean towards propagation for restoration and population augmentation and trying to recover some of the populations that we've lost through human impact. So if we get to the point where we're not seeing mussels recover at a fast enough rate and we kind of move towards captive propagation, do you have recommendations, or do you think there'll be recommendations on how can we best do that and maintain a strong genetic diversity?
Yeah, this is great questions. And yeah, we have a lot of discussion about propagation and genetic diversity and how we best practice propagation. And then I think we need to coordinate with the propagation facility and a geneticist, and at least understand what we are dealing with those rare species or species of the concern. A lot of, yeah, so we want to have some kind of, like, at least like stat book to just make sure which species, which individual, which genetic variations are propagated. So next time you're propagating, we should have a different kind of genetic variation, something like that. And then they might not have one single guidelines, right? Because so based on my study, this is a very common species. And then I don't know the trend. So the one single female has high levels of multiple paternity applies to rare species, because maybe rare species, absolute number of males in a habitat is smaller than common species. So female maybe have very small number of paternal input. It's very difficult to make guidance about, you know, common species and then rare species. But yeah, so I guess my wish is more like, yeah, we should at least collaborate with propagation facility and a geneticist to at least understand what we are trying to manage through propagation and in genetics. I don't know if that makes answering your question.
It does. Thank you so much for that. I appreciate it.
And we have a question in the chat from John Griffiths. He wants to know, do labs that propagate mussels ever do exchanges in order to improve genetic diversity?
Yeah, that's also good questions. And I don't know they are, so I don't know, I don't know what they mean by exchange, but it sounds like bringing a lot of individuals from a lot of different places so that we can kind of artificially increase genetic diversity. And we try to not to do that most of the time just because we don't want to kind of mix genetic diversity from kind of distantly related populations. There's an example of outbreeding depression. So if you are kind of putting new genetic variants in different place, that actually have negative impact because those variants might not be adapted to those specific habitat. So we always we need to kind of caution about how we bring those females to improve or increase genetic diversity by not moving animals among sites.
So I've got a question. So in Illinois, in a kind of a typical small river, so, you know, large enough that someone can maybe paddle on it, but not large enough you're going to have motorboats. How many different species of mussel would you expect to find if you have a high quality stream system? Like how many can coexist together?
Yeah, so it's a really depending on place, but usually you can find 10 to 20 species, maybe more, depending on place. Like Kankakee River, and they have a pretty high diversity of freshwater mussels. So you can find, I don't know the exact number, but I think you can find about 30 species of freshwater mussels in the Kankakee River.
Hey, Ken, it's Phil. How's it
Hey.
going? So it's interesting hearing you talk about the changes in the allelic diversity. So I'm wondering, kind of in a situation where a stretch of river has been restored to a degree, would you say that it would be probably better off protecting that diversity if you're like transplanting a population from elsewhere versus trying to create a new population just from captive stock?
Can you repeat it again?
Yeah, so like in a situation where we have a restored river, a stretch of river that doesn't have a certain set of mussels that we'd like to see there, and we're impatient or there's a block or something like that preventing mussels from moving in naturally, would it be a better practice, do you imagine, or would this take more information to say we would be better off taking some population and transplanting it ourselves, or would it be better off coming and starting a new population just based off of captive breeding? If that makes sense.
Yeah. So depending on those animals, either translocated from or propagated from, I always stick with animals. If you move animals or if you reintroduce animals, the animal itself should come from the same system. So we don't want to, like last question too, but we don't want to, cross mix any genetic variation or in the new population, just because there may be negative consequences. So yeah, I think your question is it better to use either the relocated animals or juveniles to create new population in unoccupied habitat?
Right.
It's, maybe we can do some experiment, right? We bring new adult mussels from, let's say, upstream of that upstream to the unoccupied habitat, and then see how they survive. Because adult mussels has higher tolerance to those environmental factors compared to juveniles. Juveniles are more sensitive. Or you can do either way too, because you need to have juvenile to survive or to create populations. But I guess we can do some experiment to do that.
Sounds great. Let's do it.
Okay, and there's another question from Mary Vireg. In prairie restoration work, we limit the geographical location from which we collect plant seeds to 50 miles from our restoration site. Is that analogous to the answer you gave to the question of exchanging genetic material from different propagating labs?
Yeah, yeah, it's a very similar to that. I usually say it's in the same river system. So if you said DuPage River, so if and then you want to relocate or reestablish new populations, you want to collect animals from same river system, not from, let's say, Rock River or Kishwaukee River or something like that. So there are a lot of genetic evidence showing that freshwater mussel has a lot of genetic structures, and then those structures associated with river systems, because it makes sense because fish move through the river systems. So if rivers are not connected to each other, or fish cannot move to one place to the other stream, those mussels are genetically isolated each other. And then the danger of bringing those animals to that new habitat is, like I say, it's like outbreeding depression, or they might not be able to survive because they are not adapted to this habitat. So yeah, so most of the time we recommend to take from the same system and then place to the same place, same system.
Okay, so we got one more question from John. Do you plan to expand your research to other river sites in the future? And how did you choose your current sites?
Yeah, so I'm kind of expanding sites, and then it's really depending on the project. So my field sites, field research, I selected those 3 sites, kind of based on collaborators and then kind of land use. So the Poplar Creek is more like urban settings, so there's a lot of residential zones. But in the Kishwaukee River in the DeKalb County, the dominant landscape is cornfield and soybean field. And if you go to Nipissing Creek, it's kind of mixture of agriculture and then forestry. So I wanted to know how those land use change freshwater mussels community and then population dynamics. But for genetic research, I didn't talk about that, but that one part of research I'm working on is kind of the population genetics of wide distributed species. So that species is distributed from Minnesota to the Indiana to Missouri. So I have not the site, but I have a collection from those localities across those states. So it's really depending on next my project, especially for genetic project or any lab research.
Great. Well, thank you, Dr. Inoue, for your wonderful presentation this evening. And thanks to all of you for joining us tonight. I hope that at least some of you will be able to make it back in 2 weeks on Wednesday, October 27th, when we will be joined by Mackenzie Sorsen, who is going to talk about her recent work looking at the distribution of another mollusk, the invasive Asian clam. So again, thank you all for coming and have a wonderful evening.